Wistar, of course, has helped develop multiple vaccines, and none is really more famous than Stanley Plotkin's rubella vaccine. So that vaccine was developed as a live attenuated or weakened vaccine by passage. And what that means is you grow the vaccine and move it through different cell lines or animals in order to weaken its ability to grow in human hosts. And then at some point you do go back and test that this growing and adapting it to a different place made it weak and was then safe for testing in human beings. And so that's one of the mainstays of our traditional vaccines. That would be measles, mumps, rubella, as just traditional examples that we've all heard of. And of course, the live polio vaccine. Another is killing products, and that's the inactivated Salk polio vaccine, for example, or many of the flus, where we kill them, and with chemicals or heat, for example. And those have advantages, especially the live, in that they can give very long-term immunity, but they are very, very slow, usually taking decades to develop, and that won't serve us well to test for safety in this current outbreak. And the killed have similar time frame limitations, and that it takes a very long time to produce them, ensure that they're completely killed at enormously safe levels, and can be delivered to people. So those are two of the most traditional types that we've had a lot of success with. So the difference between these older technologies, live or killed, and I would put under killed even pieces, protein pieces, is that what Wistar moved forward very rapidly with funding from global institutions such as CEPI and the Department of Defense, the Coalition for Epidemic Preparedness Innovations, was nucleic acid based, or basically information based, where what we really deliver is not a virus at all. We deliver a piece of information that instructs the person's body when they take it to build an artificial part of the virus and inside their body. And by doing that, we bypass all this attenuation. We know it's non-live, it can't grow, it can't spread, it's locally administered and locally produced, and so it has all the conceptual safety advantages that normally take decades to reach. Plus, since it's produced inside a person, it can have the theoretical immunology associated with a live approach. And so we deliver information that instructs the body to produce an artificial virus protein, in this case spike, and that spike protein becomes targeted by the immune system, making antibodies and T cells.
We're sort of at a time in human history where we understand a lot about human health, but we don't fully grasp all aspects of the immune system. So there's no silver bullet regarding vaccine approaches. So vaccine scientists have created many different types of technologies, and it's still really an active area of research. However, it turns out we've learned a lot, and we learned that the immune system is quite robust and can be stimulated into action in many different ways. So vaccine researchers have developed alternative technologies, which was described by David a few minutes ago, to sort of exploit this feature of the human body. And each one of these approaches, whether it's killed or live, or one of these newer approaches, the nucleic acid approaches, each require sort of unique set of experiments to evaluate the vaccine candidates. However, in the end, all vaccine candidates must demonstrate some induced immunity in preclinical and clinical settings for broader use in the human population.